tell. All right, so good afternoon to everybody. My name is Pierre-Olivier Gaudreau, and it's a great pleasure to be hosting the first conference of the 2021 Queen's Cancer Research Institute monthly seminar series. I would first like to thank everybody who contributed to the reactivation of this series, with special thanks to Maureen Hobbs, Nicole Lawrence, Majuri Koti, Vishal Goyali, and David Berman, along with all colleagues who provided suggestions and contacts for our upcoming speakers. Secondly, I would like to thank you all for attending this seminar. Please note that this presentation will be recorded and that it will be available on the QCRI website. So I would please ask you to make sure that your microphone is on mute during this presentation. There will be approximately 10 to 15 minutes being allowed for questions at the end of this presentation. If you do have a question, may I please ask you to write it down simply using the chat function and addressing the question to me, so I can basically act as a moderator at the end of the presentation. So without further introduction, I'm very excited to present a friend and colleague today, Dr. Triparna Sen. Triparna completed her PhD in cancer biology at Jadavpur University in India, and then joined Dr. Lauren Byers' lab at the MD Anderson Cancer Center as a postdoctoral fellow, before getting appointment as assistant attending at the Memorial Sloan Kettering Cancer Center since April 2019. Triparna's outstanding work in small cell and non-small cell lung cancers has been repeatedly recognized as of late. Besides some of her work being published in prestigious journals, including Nature Communications and Cancer Discovery, in 2020, she received a Young Investigator Research Award in Translational Immuno-Oncology from the Lung Cancer Foundation of America and the International Lung Cancer Foundation. Moreover, she received the Under 40 in Cancer Award, Rising Stars and Emerging Leaders. So without further introduction, it's a great pleasure to welcome Triparna and her presentation entitled Novel Insights into Recalcitrant Subtypes of Lung Cancer. So Triparna, the microphone is yours.
Thank you so much, Pierre. I'm really glad to be here with you all today. Thank you so much, Pierre. You've been a wonderful colleague during our time together at MD Anderson, and I'm really happy to be presenting this work today. So as you can see from my topic title, I'm going to talk about multiple subtypes of lung cancer that have been the hardest to treat and have the worst prognosis. So the work that we have done to understand the biology of these subsets and also some of the therapeutic leads that we have got. So as I'm sure this audience is aware of, lung cancer is the largest cause of cancer-related mortality globally. This statistics is about 6 years old, but what you can appreciate is that about 1.8 million people have new cases of lung cancer per year globally. And there are 1.6 million deaths that happens with lung cancer disease globally. In the US, the statistics is very similar. Non-small cell lung cancer alone is the largest cause of cancer-related mortality in the US. And non-small cell lung cancer is about 80 to 85% of lung cancer. Small cell lung cancer, which is the 15% of lung cancer cases have this subtype of lung cancer, it is the 6th most common cause of cancer-related mortality. But even with that, we have over 30,000 deaths per year in the United States alone. So it is a huge public economic burden, this disease. So today I'm going to talk about two specific types of lung cancer that we have seen to be harder to treat. I'm sorry, there's a typo in my slide, but my recalcitrant lung cancer number one would be the STK11 and KEAP1 co-mutant lung adenocarcinoma. And in the second part of my talk, I'll be talking about small cell lung cancer. So for a long time now, the treatment decisions for lung adenocarcinoma has mainly been based on the driver mutation status of the tumors. When the patient comes to the clinic, we look at the next generation sequencing for those patients. We identify driver mutation in either EGFR or KRAS, and therapeutic decisions are mainly based on these driver mutation status of these tumors. And that has been the case for a very long time now. However, in the last few years, we have understood the importance of co-alterations, like loss of function mutation in STK11 and KEAP1, which would be sort of the central theme of my talk today. But we have understood that having these co-alterations like LKV loss or KEAP1 loss along with KRAS actually decides how the cancer would respond to treatment, whether they would be resistant to immunotherapy or chemotherapy, and their metastatic proclivity. And this will be clear with the next study that I'm showing. So this was a study that was led by a group at Memorial Sloan Kettering Cancer Center, where they looked at the mutational status of over 1,000 metastatic lung adenocarcinoma cases. And they looked at the mutational status and their co-occurrence status. Depending upon the mutational status, they associated a risk score. So a low risk score means a higher overall survival, and a high risk score means shortest overall survival. So these mutations were supposed to be a prognostic marker for these patients. What you can take away from this slide is that having a co-alteration in STK11 and KEAP1 actually predicted shortest overall survival for these patients. Hence, they fell into the highest risk score. And what you can also see that a lot of these patients did not have a driver mutation in KRAS. So they happened irrespective of the driver mutations, and they were still predicted worse prognosis. So a little bit about the genes that I'm talking about. So serine threonine kinase 11 or STK11, it encodes for the protein named LKB1. So during my talk, I'll go back and forth between STK11 and LKB1. And it directly phosphorylates and activates the AMP kinase family member. So it plays a huge role in the energy homeostasis of lung cancer. And STK11 mutations are present in about 33% of lung adenocarcinomas. When we come to KEAP1 is a negative regulator of NRF2 that activates the expression of 200 antioxidant response elements containing genes. And KRAS KEAP1 mutant lung adenocarcinoma has been shown before in this reference that I'm showing before to be dependent on glutamine metabolism. And KEAP1 mutation is seen in about 17% of lung adenocarcinomas. In our study, we looked into 1,235 metastatic lung adenocarcinoma cases that have undergone next generation sequencing with MSK-IMPACT platform. What we can appreciate is that 10%, so even in this relatively smaller cohort of patients, about 10% of them had STK11 and KEAP1 co-mutation, which means it is a very frequently occurring co-mutation. And interestingly, 46% of these cases did not have a KRAS mutation, so they had a KRAS wild type status. When we go into the overall survival data, what you can appreciate is that having an STK11 KEAP1 mutation alone decreased the median overall survival by half, like it decreased from 26 months to 11 months, so less than half. And having a driver KRAS mutation, of course, made it worse. And then the median overall survival decreased to about 6 months. So we hypothesized from the literature and from what we saw in our data set is that a loss of function mutation in STK11 and KEAP1 cooperatively promoted lung cancer progression and worse prognosis. And we believe that it is through a synergistic axis, which is independent of the driver mutation KRAS status. Now, we wanted to interrogate these mutations in the lab. For that, we made multiple preclinical models. And H358, which already had a KRAS mutation, the KRAS G12C mutation, and we selected another model, which is H292, which had a wild type KRAS status. What we did was that we created 4 genotypes of these models. The non-targeting control is they are wild type for STK11 and KEAP1. STK11-KO is just the knockout of STK11 alone and knockout of KEAP1 alone, which is the KEAP1-KO. And this purple thing here, which is DKO, has a co-alteration or co-mutation of STK11 and KEAP1. We took these models and we interrogated it in multiple ways. We first looked into the biological characteristics, and I'll talk about proliferation for this talk. We looked into the unique gene expression programs by bulk RNA sequencing, and we also wanted to look into selective dependencies or identify novel therapeutic targets, for which we conducted a CRISPR-Cas9 druggable genome screen with over 400 genes in the screen. So this work, I would acknowledge the person who actually did the work first. So this is the work that I'll be showing you for this part of my talk, has been by a stellar graduate student who's recently got her PhD degree, Corinne Wall-Hater. So what Corinne did was she made these models in the lab. So what you can appreciate from the Western blots is the single mutations had the knockouts. So LKB1 is the protein for STK11. So the STK11 knockouts had a loss of LKB1, and also the KEAP1 mutants alone had a loss of KEAP1. The double mutants, of course, had a loss of both these two genes and proteins. And since KEAP1 is a negative regulator of NRF2, you can see NRF2 being upregulated in these models when you knock out KEAP1. So these models were very successfully generated. When we looked into the proliferation status through crystal violet staining, and then we quantified the proliferation through a colorimetric assay, what we could appreciate is that with STK11 and KEAP1 knockout alone, the proliferation of these models did increase. However, the proliferative potential significantly increased when you had a co-mutation of STK11 and KEAP1. So we were seeing what we are seeing in patients, that these tumors are more aggressive, more proliferative. Now, she also did in the other way. She overexpressed STK11 and KEAP1. So she chose A549 for these models, which has a de novo loss of KRAS, STK11, and KEAP1. And she brought back STK11 or KEAP1 expression, as you can see in these models, that she was able to overexpress either LKB1 or KEAP1 in these models with a concomitant downregulation of NRF2. When we injected these models into the mice, what you can see here, that the ones which have an STK11 and KEAP1 co-mutation, which is this blue and the red line here, they had a shorter survival in mice. However, when you added back KEAP1, you significantly prolonged the survival of these mice. So adding back could actually decrease the worst prognosis in these models. So when we did bulk RNA sequencing of all 4 genotypes, the non-targeting control, the single mutants, and the double mutants, what we found was that Pheroptosis was one of the 2 topmost upregulated gene sets in the double mutants. And I'm showing you the particular genes that we have seen. So glutathione metabolism, or glutamine metabolism, as we have seen in previous literature, it came up to be the topmost upregulated pathway, which is sort of a proof of concept for us. We knew that the screen worked. So what you can see here, that multiple genes that are involved in Pheroptosis regulation or Pheroptosis evasion was actually upregulated in the double mutants. For those of you who do not work with Pheroptosis on a regular basis, Pheroptosis is a non-apoptotic form of programmed cell death. And it depends upon the lipid peroxidation of the cells, which means you can induce Pheroptosis by increasing the lipid peroxidation in these models. And GPX4, one of the genes, which is a master regulator of Pheroptosis, controls this lipid peroxide levels in the cells. It's a relatively newer phenomenon. It was identified in 2012, but even in this last less than a decade or so, we have understood the importance of Pheroptosis, not only in cancer, but also in other malignancies. So next, we wanted to see whether these double mutant models are really resistant to Pheroptosis. So we inhibited GPX4 with RSL3, which is an inhibitor for GPX4. What you can see here is that the non-targeting control and the keep one single mutant alone was extremely sensitive to ferroptosis induction with GPX4 inhibition. However, STK11 knockout made them more resistant, and the double mutants were extremely resistant to ferroptosis induction with GPX4 inhibition. Now, a very good assay to look at ferroptosis is body PC11 assay, which measures the lipid peroxide levels in the cells. So for this, we chose two models, H358, and for this experiment, we chose the wild type model, which is wild type for STK11 and KEAP1. And what you can see that with GPX4 inhibition with RSL3, you could cause complete cell death, a ferroptotic form of cell death when you treated it with RSL3. So these wild type models were very sensitive to ferroptosis induction. However, A549, which has a de novo loss of STK11 and KEAP1 mutate, STK11 and KEAP1 were resistant to cell death induced by ferroptosis induction. So these cells were indeed resistant to ferroptosis. When we added back LKB1 and KEAP1 into these models, we could then induce ferroptosis. So the cells needed both of these two genes to be loss of function mutation in order to be resistant to ferroptosis. Even adding back one of the genes made them sensitive to ferroptosis. Now, next, we conducted a screen. And for this, I would like to thank Fatima Odin, who's a spectacular research technician in the lab. She conducted the CRISPR-Cas9 screen with the druggable genome library, for which we transduced pool 1 and 2, which includes more than 1,400 genes with 4 guides per gene into this screen. And we did it for all 4 genotypes, the wild type control, the STK11 and KEAP1 knockout alone, and the double mutants. We continued the cell lines. We continued to grow them for 16 doublings, after which we measured the guide RNA dropout by next generation sequencing. What you can see here is that with RNA-seq data, what we first noticed was that a particular gene was upregulated, which is SCD1, which is a sterile CoA desaturase, which was one of the topmost upregulated genes when we analyzed the bulk RNA sequencing data. However, interestingly, even in the CRISPR screen data, SCD1 was seen to be the topmost dropout gene. So which means, in other words, these double knockout cells actually needed SCD1 in order to proliferate. So dropping them out was actually deleterious for them. So a little bit about SCD1, I'm going back to the ferroptosis pathway. So SCD1, it actually changes saturated fatty acids to monounsaturated fatty acids, and it inhibits lipid peroxidation. So it is one of the ferroptosis evasion genes that we saw to be upregulated, and also a dependency in these double knockout models. Now, we really wanted to see whether SCD1 is essential for these genes to survive or not, an indication that we got through the CRISPR screen. So we knocked out SCD1 in our double mutant models. So what you can see here is that in our single mutant, so we knocked out SCD1 in all our 4 genotypes. In the non-targeting control, we did not change the viability when we knocked out SCD1. So SCD1 knockdown did not really change the viability of these non-targeting control. For STK11, we were able to see some form of cell death. Even in KEAP1 knockout, we were able to see some form of cell death. But look at the shift here. At day 1, 93.4% of the cells were alive in the double mutant models. However, with SCD1 knockout, it was a time-dependent decrease in cell viability, which decreased from 93% to 15% at day 12 with SCD1 knockout, whereas the safe targeting control, as you can see here, we did not change the viability. So SCD1 is essential for the STK11 and KEAP1 co-mutant cells to survive. Of course, we wanted to test this in vivo. So we injected the non-targeting control and the double knockout cells into the mice, and we treated them with an SCD1 inhibitor. What you can see here, that SCD1 inhibition, pharmacological inhibition of SCD1, did not really change the tumor growth pattern in the non-targeting control. However, we were able to see a significant delay in tumor growth in the double knockout cell lines when we inhibited SCD1. We also saw a same survival benefit when we treated these models, As you can see here, with the vehicle-treated mice, they had a survival of average of 70 days. However, when we decreased SCD1 or inhibited SCD1 with this drug, we could cause a significant prolongation in the survival of these mice. In fact, 2 out of 5 mice that were a part of the study had a complete response with SCD1 inhibition. So I think I've been able to convince you with this part of my talk I hope you guys can still hear me. So I hope I've been able to convince you that STK11 and KEAP1 co-mutation predicts shortest overall survival in patients with metastatic lung adenocarcinoma. It also promotes cell proliferation in vitro and tumor growth in vivo. It promotes higher expression of pheroptosis protective genes, and it's resistant to pheroptosis inducing agents. So having this co-alteration is actually deleterious for these cells. And inhibition of SCD1 prevents growth and sensitizes to pheroptosis induction. So we think this pheroptosis protective mechanism is through the SCD1 gene. And future studies will of course involve, because you could appreciate from that in vivo data that we still do not have very potent and targeted SCD1 inhibitor. So the future studies of our group is to develop specific and more targeted SCD1 inhibitor that could not only be treated in the clinic for the subset of patients, probably as a single agent, but also in combination with existing chemotherapy, immunotherapy, and also maybe in combination with pheroptosis-inducing agents like Erastin or Aracil-3, which are already in clinical trials. So for the second part of my talk, I'll talk about the second type of recalcitrant lung cancer, which is small cell lung cancer. Small cell lung cancer has been my interest for a very long time now. It is an intriguing disease. So these are the 2 major studies that have shown the genomic status of small cell lung cancer. As you can see here, this study and this study, what you can appreciate from the slide is that this cancer has a lot of mutations. Like this is a very highly mutated cancer. And this gene of this cancer almost has an ubiquitous loss of P53 and RB1. So almost all patients that have been detected have an ubiquitous loss of function mutation in P53 and RB1. Now, the treatment regimen for small cell lung cancer had been chemotherapy for a very, very long time. For almost 4 decades, chemotherapy was the only standard of care for frontline treatment for small cell lung cancer. However, immunotherapy recently got introduced since 2018 in the frontline regimen. So now small cell lung cancer patients who are unfortunately mostly diagnosed at the extensive stage disease are treated with frontline chemotherapy and immunotherapy as a combination. So it's usually either atezolizumab or durvalumab that are FDA approved right now. So at this stage, patients usually see an objective response rate of more than 60%. So most patients respond, but we see this response is very short-lived, and almost all of them relapse. And when they relapse from second line onwards, the drugs do not work as well. And we get an objective response rate of less than 20%, which leads to a very dismal overall survival rate for extensive stage disease, which is still, the median is between 9 to 13 months for extensive stage small cell lung cancer patients. And what is most alarming is that the 5-year survival is still less than 1% for the extensive stage disease. So we still haven't been able to shift the needle as much as we would like to. A little bit about the transcriptomic status. So what the field understood was that looking into the mutation status did not inform us a lot about the therapeutic regimens that we should be giving these patients, which was very unlike non-small cell lung cancer. So we looked into the transcriptomic status of small cell lung cancer, and we tried to subdivide this cancer according to the gene expression status. So what you can see here is that small cell lung cancer can be broadly divided into 4 transcriptionally driven subtypes, which is either seen as a relative expression of ACL1, NeuroG1, PAU2F3, or YAP1. So these are the 4 major subtypes sort of what the field now follows. But it's an ever evolving, and that will become more clear in the later part of my talk, is that we are still knowing more and more about the disease with more and more studies that we are doing. So just talking about what is new in small cell lung cancer. So the field is really interested and really sort of happy about having immunotherapy in the front line. This is one of the studies that I'm showing you, which is the Caspian study, which led to the approval of durvalumab in the frontline therapy. You can see that the shift is very modest. And the Empower 133 study, which was with atezolizumab and chemotherapy, the curve looks exactly the same. So for both of these studies, we could see a benefit of the median overall survival by only about like 2 months. But even that was a shift in the needle for small cell lung cancer patients, and hence these drugs got approved. But I think we can do better. We can do better than improving the overall survival by just 2 months. And when you look at all the studies that have been done, Empower 133, Caspian study, and the Keynote 604, and also looking at not only the frontline therapy, but also the second and third line immunotherapy, there is almost a 10% rule of the progression-free survival. Every curve looks like a hockey stick, and in every case, only about 10% of the patients benefit or have a noticeable increase in the progression-free survival. So there's much more work to be done in order to potentiate the effect of immunotherapy in small cell lung cancer. So with this idea in mind, we looked into small cell lung cancer, and this is a study that was published by MD Anderson, led by Lauren Byers back in 2012, where they looked into the protein expression between small cell lung cancer cell lines and non-small cell lung cancer cell lines. What they interestingly noticed was that a bunch of proteins that were regulating the DNA damage response pathway was seen to be higher, like PARP, CHEK1, ATM, and ATR. So everything which was downstream of the E2F1 transcription factor were seen to be higher in small cell lung cancer. So why do we think DNA damage response is a vulnerability for small cell? As I've shown before, small cell lung cancer has an ubiquitous loss of p53 and RB1. So if you think about the cell cycle and about the two major checkpoints, the G1S checkpoint and the G2M checkpoint, the G1S checkpoint is under the control of p53. So if you have a loss of function mutation in p53, your G1S checkpoint is now completely compromised. So there is no checkpoint. Now the cell depends upon the G2M checkpoint for its repair upon DNA damage. So the G2M checkpoint is under the control of major proteins like CHEK1, ATR, and V1. So our hypothesis was that if you could target the G2M checkpoint with either targeting CHEK1, V1, or ATR, or if you could target the repair of DNA with PARP inhibition, you could cause cell death. In our previous study with a single agent of these drugs, we have shown that CHEK1 inhibition in small cell lung cancer produces single agent effect. So it was not only higher, we not only see a higher expression of CHEK1 in the small cell lung cancer tumors, these are patient tumors, but we were also able to cause a significant decrease in tumor burden in multiple tumor models in mice. And we proposed c-MYC as a predictive biomarker for CHEK1 inhibitor treatment. The hypothesis for combining it with an immunotherapy is as follows. So if you cause a DNA damage, and we know from literature from several other studies that increasing a DNA damage can increase the immunogenicity of your tumors. And by increasing the immunogenicity of the tumors, you're able to have higher tumor infiltrating lymphocytes. Now, when you combine that with an anti-PD-L1, you can release the brakes on the immune response, and combination of these two can lead to the tumor cell clearance, which is exactly what we saw, as you can appreciate from this model. Sorry. So as you can, so this was a genetically engineered mouse model that we treated with either vehicle, olaparib, which is a PARP inhibitor, anti-PD-L1, or the combination. So treatment with vehicle, olaparib, and anti-PD-L1 alone did not do anything to these tumors, which are very aggressive. As you can see here, by 22 days, we lost all mice in all the other groups. However, in the combination treatment, we got complete tumor clearance as early as like 2 weeks. We started seeing tumor clearance by day 12. And it was sustained tumor clearance up to about 3 months that we have followed this tumor. And of course, that led to a very significant improvement in the survival of these mice. We got the same result with the CHEK1 inhibitor. So again, vehicle and anti-PD-L1 did not do anything for these tumors. So PD-L1, like we see in patients, they do not work in these mice models. So having an immunotherapy alone is not really effective in small cell lung cancer. However, CHEK1 inhibition alone could significantly decrease the tumor burden. However, when you treated these mice with a combination of anti-PD-L1 and CHEK1, you could clear mice. So I'm saying 60% of the mice had complete tumor clearance. You could no longer see the tumors. And you can see individual mice tumor burden here. Even if we did not have a complete tumor clearance, they never reached above baseline. So we were able to control these tumors very effectively. Of course, the next thing we wanted to look into the immune status of these tumors. So we resected these tumors at day 21 and did a multicolor flow cytometry to assess the anti-tumor effect. So I'm just summarizing. This was like a 20-color flow. So I'm just summarizing the data that we got. The first thing that we noticed was that there was a significant increase in the memory effector CD8 positive T cells, which were denoted here by CD44 high and CD62 low. So there was a significant increase in memory effector CD8 positive T cells. And that is also clear with the sustained response that we get with these drugs. So when you treat it either with a combination of PARP and anti-PD-L1 or with CHEK1 and anti-PD-L1, the response or the tumor clearance is very sustained. 3 to 4 months, the tumor never comes back, which is also shown with the increase in the memory effector CD8 positive T cells. What we also noticed was that these tumors had a higher total T cell infiltration, which is the CD3 positive T cells. But the best part or the interesting part for us was an increase in the cytotoxic T cell or the CD8 positive CTLs that infiltrated these tumors, significantly higher than either the vehicle or the single agent. So this was very encouraging because we are actually recruiting CD8 positive T cells into these. So in the next question, we obviously asked that is this response truly CD8 T cell mediated, for which we depleted CD8 positive T cells in these mice models. What you can see here in our IgG control, where we depleted T cells, you could see, so this graph right here, I'm showing you that we were able to cause CD8 depletion effectively. So in the CD8 depleted population, you do not get any change in CD8 positive T cells, of course, because CD8 is depleted here. However, in the IgG control, like we saw in our previous model, the combination actually significantly increased CD8 positive T cells. But CD8 depletion, interestingly, when you look at the tumor growth, it actually reversed the tumor growth. So you were not seeing tumor clearance when you take out CD8 positive T cells from the equation. So we concluded that this response or the tumor clearance was at least in part mediated by the cytotoxic T cell infiltration. Now, when we treated these cell lines with either the CHEK1 inhibitor alone, which is I'm showing you here by the single agent, or with PARP inhibitor alone, we saw micronuclei formation in these cells. Now, micronuclei formation has been shown in the past decade or so, like we know it from literature that micronuclei formation actually increases the cytosolic DNA in your cells, and that activates the cGAS-STING pathway. So we were interested to see, since we are increasing the micronuclei formation, are we activating the cGAS-STING pathway? That was the next question that we asked. So what you can see here, either with olaparib, which is the PARP inhibitor, or prexasertib, which is a CHEK1 inhibitor treatment alone, we were able to increase every notable player in the cGAS-STING pathway. So phospho-STING, phospho-TVK1, cGAS, and phospho-F3. We saw an upregulation of these regulators of the cGAS-STING pathway in our cell lines, which were treated with a single agent of these DNA damage response inhibitors. In our tumors that we resected from the mice, again, we saw the same thing. Just with the single agent of olaparib or prexasertib treatment, we were able to activate the cGAS-STING pathway in all our models. So again, we went downstream since we were seeing ERF3 being generated, and I'll talk about the pathway in a little bit when I summarize my study. We looked into the type 1 interferons, which we know from literature to be activated by the ERF3. We looked into IFN alpha, IFN beta, and we saw that IFN beta was specifically upregulated either with treatment of a check1 inhibitor or a PARP inhibitor. And we also saw increase in the gene expression of chemokines like CXCL10 and CCL5, which are antitumorigenic. So next question we, of course, was, is TGAS and STING important for this response? And the answer was yes. So we knocked out CGAS and STING in our models, and knocking out CGAS actually completely reversed the tumor clearance. So if you can see here, the green line, so the control is the black line, and the green is the scrambled control for our CGAS knockout. And when you treat it with a combination, again, you were getting complete tumor clearance. But when you knocked out either CGAS or you knocked out STING, you were no longer getting this anti-tumor effect. So this model, it needed intact CGAS and STING in order to have this anti-tumor effect. So I'm just going to summarize the pathway that we proposed in our study. So when you treat small cell lung cancer models with a PARP inhibitor or a CHEK inhibitor, you cause double-stranded DNA breaks, which we saw from either increase in gamma H2AX or the micronuclei formation. This leads, and we have confirmed that, upregulation in the cytosolic DNA. Now, with cytosolic DNA upregulation, you get an activation of the CGAS-STING pathway. You get recruitment of ERF3 and TDK1. Now, ERF3 phosphorylation actually leads to the transcriptional upregulation in our models of IFN beta, which then either leads to PD-L1 upregulation, we have shown that in our study as well, and also an upregulation of CCL5 and CXCL10, which leads to T-cell recruitment and also decrease in T-cell exhaustion. Now, when you come in with an anti-PD-L1, you are able to then clear tumors in these mice. Now, why are we really interested or really sort of excited about this study? There are several DNA damage repair inhibitors that are in trials for small cell lung cancer, either as a single agent or in combination with chemotherapy. I'm showing you CHEK1, ATRNV1 here, and PARP inhibitor here. And what's most encouraging is that we right now have combination clinical trials, either with a PARP inhibitor and with immunotherapy. So these are ongoing clinical trials. We do have previous results from Mediola trial, but there are other trials with advanced designs that are currently being treated in patients for extensive stage small cell lung cancer. And we are really interested to see whether the response that we got in our mice actually get recapitulated in the patient or not. And that we think would really be a game changer for immunotherapy in small cell lung cancer. For the last 5 minutes, let me see how much, yeah, for the last 5 minutes, I'll just talk very briefly about where we are taking our studies next. So as I said in the beginning, we are trying to understand small cell lung cancer, which we believe is underrepresented in a lot of studies that we have done. We have no TCGA data because it is not a surgical tumor. So there is no larger study that are being done with small cell lung cancer, and hence our knowledge about these tumors remain incomplete. So for that, we have joined forces, and I'm showing you the people who actually did the work. We joined forces to look at small cell lung cancer clinical samples at the single cell level, because we think that this is an extremely heterogeneous tumor and it is also plastic. So we looked at these clinical samples at the single cell level. So I'm just showing you sort of the graphical of how we continued the study. Is we took all cells and we sort of had non-small cell. We collected tumors from non-small cell and small cell lung cancer. We selected with these markers where we were able to isolate just small cell lung cancer tumors by looking into the neuroendocrine status of these cells. So we were able to then see small cell lung cancer within this larger lung cancer atlas. We were able to identify small cell lung cancer cell lines. Not only that, we were able to identify the 4 major transcription factors that we showed before in each of these, in the models that were actually small cell lung cancer. I'm going a little bit again into the complexity of the small cell subtype, because I think you need to understand this, that it is a very heterogeneous cancer, and there is a shift between one subtype to another, which makes treatment so tricky for small cell lung cancer. So as I said before, in the bulk transcriptomic data, we had 4 major subtypes: ACL1, NeuroD1, PAL2F3, and YAP1. We were able to detect, so ACL1 is the most prevalent small cell subtype, as you can see here. More than 50% of the small cell lung cancer patients or tumors that were assigned in this study had ACL1 driven. So in our study, of course, the majority of the cells had a higher expression of ACL1. With the second subset was NeuroD1, and then we got PAL2F3. We unfortunately did not have a YAP1 representation in our models. So when we looked into the pyramid graph of these cells, just cell-wise, you can see that you can differentiate most of these cells into one of these 3 major transcription factors. However, there are many cells in between that either have both expression of the both ACL1 and NeuroD1 markers. And now currently in our mouse study, which was published by Huntsman Cancer Institute, what we also saw is that upon treatment, small cell lung cancer changes from ACL1 to the NeuroD1 subtype. Now, when we looked into the small cell lung cancer clusters, what we saw, there was one cluster that was seen across all subtypes, and it had a very stem-like property. This cluster had SOX4-mediated genes upregulated. It had stem-like gene-mediated, stem-like gene signature upregulated. And also, interestingly, it had metastatic signature upregulated. The gene that we identified to be the highest in this cluster, in this prevalent cluster in small cell lung cancer, was PLC gamma 2. Now, PLC gamma 2 is seen across small cell subtypes, and we believe is a metastatic driver for small cell lung cancer. And why I'm saying this is because when we looked into either the lung tumors or the tumors that we got from the metastatic sites, we saw an increase in PLCG2 when you see the distant metastatic sites like liver, lymph nodes, pleural effusions. You saw an increase in PLC gamma 2. Now, now we wanted to investigate this in the lab. So we upregulated PLC gamma 2 in a model that did not have the de novo expression. And we also knocked out PLC gamma 2 in the DMS114, which had a higher expression of PLC gamma 2. What you can see here is that upregulating PLC gamma 2 actually led to a higher migration and invasive potential in H82. And likewise, when you decreased PLC gamma 2 or knocked out PLC gamma 2, you were able to decrease the migration and the invasive potential in these models. And interestingly, we also saw that cells having higher PLC gamma 2, and this was measured by immunohistochemistry, had a worse prognosis in small cells. So actually, PLC gamma 2 was predicting the worst phenotype for small cell lung cancer. When we looked into the immune population, we saw a profibrotic microphage population that was seen to be higher in small cell lung cancer and also higher specifically in the metastatic sites for small cell lung cancer. So of course, this is very much a work in progress. We're still looking into the questions. There are many more questions to be answered here. The first question is PLC gamma 2 high population necessary for tumor progression or not? For which we have to do in vivo studies, of course. How is PLC gamma 2 high cells distributed in tumor? What is the neighborhood? What are the cells that is interacting with? So there is much to study about the tumor microenvironment of these PLC gamma 2 high cells. And this unique the fibrotic cluster that we got in our small cell lung cancer immune cells, are these tumor promoting or restricting? So we have to do more targeted studies for that. And we also want to see the crosstalk of other immune cells and the tumor cells. Like, is the PLC gamma 2 high cells talking to this fibrotic population or not? So there are many, many more unanswered questions, but we're very excited about the initial study. So I just wanted to show you some data. And just the few conclusions, this is my last slide. So we have undertaken an effort to understand the biology and identify therapies for the most recalcitrant subtypes of small cell lung cancer. And the new toolbox that we have sort of generated in our group includes single cell profiling, CRISPR screens, and comprehensive immune profiling and expression profiling through bulk RNA sequencing, with which we think we can understand the biology of these diseases better. What I showed you in the first part of my talk is that STK11 and KEAP1 co-mutant lung adenocarcinoma markedly induce and are uniquely dependent on anti-ferroptotic pathways. In the second part of my talk, I hope I've been able to convince you the small cell subtypes, they define a key transcriptional regulators and they might provide a roadmap for clinical translations, and have shown you that you can treat these small cell lung cancer cell lines either with a DNA damaging agent, either with a PARP inhibition or with a CHEK1 inhibition, and potentiate the effect of immunotherapy in small cell. Single cell profiling, which is very much again a work in progress, we have shown a rare PLC gamma 2 high population, which has stem-like features and which we think has unique metastatic characteristics, and a profibrotic macrophage population we have identified, which still has unknown significance to us, and for which we are actively looking for that into the clinic. Sorry, in our preclinical models. We're a long way from the clinic. With that, I'd like to acknowledge, so as you can understand, it is a team sport. Research is always a team sport. So I would like to thank everyone who contributed. We have a fantastic clinical team. We have fantastic people in the lab, grad students, postdocs, and technicians, and other collaborators from other institutes who have either generously donated their cell lines, their animal models, and this work would not have been possible with our collaborators from pathology and the single cell sequencing core. Of course, the funding institutions who have supported our work, I have to thank them for the support that we get, because we need that support. With that, I'm happy. I hope I have left time. I have left 10 minutes for questions, and I'm very happy to take questions. Again, thank you so much for having me here today. And thank you, Pierre, for the invitation.
All right, Triparna, thank you so much for accepting to present and for this compelling presentation. Indeed, we have about 10 minutes left for questions. I received a few already. First, we have a question from Dr. Ran, which refers, I believe, to the series of experiments which were done with animal models and the CHEK1 inhibitors and anti-PD-L1. He was asking, how long did you treat the immune competent mice with anti-PD-L1 antibody? Mm-hmm. And the other question was, did you observe the anti-drug antibody response in mice?
I'll answer the first question, and then I might need clarification about the second. For the first question, so these mice, the genetically engineered mouse models that we treated, had a co-alteration, had a loss of function in P53, RB1, and P130. So I just want to explain the model a little more. These were genetically engineered and had a loss of P53, RB1, and P130. We engrafted the mice, and then when they reached a tumor burden of about 100 cubic millimeter, they were treated with anti-PD-L1 for 3 weeks, and the treatment was 300 micrograms per mice per week. So once a week treatment with 300 micrograms of anti-PD-L1. So that, I hope, answers the first question. Pierre, could you clarify the second question for me?
I can repeat. I don't have any additional input, but the question was, did you observe the anti-drug antibody response in mice? I believe Dr. Ran is still with us, if I'm correct.
Yeah.
Yeah, I want to ask, because the anti-PD-L1, this antibody is generated from the rat. Mm-hmm. So you inject the antibody from rat into the mice, it may be immunogenic, right?
Oh no! So we got the mouse homolog for this antibody. So we did not see any effect on the mice or any sort of toxic effect on the mice with the anti-PD-L1 treatment, because this was generated for in vivo studies in mice. So okay. We did not see that effect. No.
Okay, thank you.
You're welcome.
Right. Next, we have a question from Dr. Craig in regards to the lung adenocarcinoma project. Mm-hmm. And the question is, what was the source of druggable sgRNA library, and how many distinct pathways were identified in dropouts?
Mm-hmm. That's a very good question. So this druggable genome library is what we made in the lab. So there are 4 pools for this library: pool 1, pool 2, pool 3, and pool 4. And we used pool 1 and 2. So how this library was designed in the lab, this is an in-house, but I'm happy to share it with anyone who is doing a CRISPR screen right now. It's published, so you can look at the genes of interest. So in our screen, we included pool 1 and 2, which had over 1,400 genes of interest. And how these genes are selected is that pool 1 would have something which has an FDA-approved drug. And then we go down having a protein downstream that has an FDA-approved drug. We saw several pathways that were dropped out. Of course, NRF2 targets, as you would expect, because you have the TEAP1 knockouts. We got phoroptosis. We got a lot of cell death pathways that we have seen, but we had ruled out those as being not essential for these models to survive. So I think the main pathways that we saw were to do with metabolism. A lot of glutamine metabolism genes were seen but we already knew that these are therapeutic targets for the subtype. And then we found, of course, phoroptosis, NRF2 targets, and a lot of cell cycle and cell death. Those were the major.
Thank you so much, Triparna. I have one additional question from the audience. This is from Dr. Chin Sang. Nice work on the DKO on phoroptosis. Since the proliferation can be suppressed with either overactivation of STK11 or KEAP1, and since STK11 is an activator of AMPK, can you treat the DKO cells with AMPK activator like AICAR to suppress the phoroptosis resistance?
That's a brilliant question, actually, because ICAR we have seen to be actually also one of the screen dropouts. So I would think yes. I haven't treated it in the lab, but if I were to make a guess, yes, I think we would be able to get the response, because ICAR was seen to be one of the genes that were seen as one of the dropouts, which were regulated by the AMP kinase pathway. So I haven't done the study in the lab, but my guess is yes, we would be able to see a decrease in proliferation even with an ICAR.
Are there any additional questions from the audience? If not,
I have a question. It's Myron Suchak. How are you? My question here is looking at the tumor microenvironment, and it has been well known that the tumor reprograms the stromal cells to become a hallmark of immunosuppression. And if that's the case, then how would you go about translating this in the clinic in terms of treating patients with small cell lung carcinoma? And I'm thinking here maybe to initiate this abscopal effect using radiation, where you can damage some of the, very precisely damage the tumor, releasing the tumor antigens, and then come back with immunotherapy, for example, with anti-PD-L1 antibody or anti-CDL4 antibody to really eliminate that. What are your thoughts?
Yeah, you're very right, because as tumor microenvironment, of course, plays a huge role. And with small cell lung cancer, we haven't, now we have looked more into the IHC, and also we are now doing MIDI, which looks into like many markers, both in the tumor cells itself and also in the microenvironment. It's like an immune desert. So we don't see any cytotoxic T cells or anything that would predict a better response in the microenvironment. So our hope is with the therapies that we're trying to treat or trying to test in the lab, either with a DNA damaging agent And today I did not have a time to talk about it, but also with epigenetic modifiers. We are looking into the microenvironment and seeing if these would actually help in recruiting T cell to the environment, not just tumor intrinsic, but also in the microenvironment population, whether we are able to induce an effect. But of course, as you said, with radiation or with chemotherapy, that effect in combination with anti-PD-L1 or with our drugs would be probably the most effective way to go. But I don't really have a straightforward answer to your questions because we're still looking into that. But yeah.
You looked at adenocarcinoma of this non-small cell lung, but there are other subtypes, for example, the squamous carcinoma, as well as the large carcinoma. The squamous is the epidermal type, and the large cell is any part of the lung, mainly in the outer regions. Do you see very similar effects of these particular cells compared to the adenocarcinoma, which is more glandular?
We haven't really done the comparative study. There is a different project in the lab which looks into transformation. And there we are seeing squamous carcinoma evolving, but we haven't done the study like we have done in adenocarcinoma to look into the driver mutations and how that changes prognosis. So we haven't done a comparative study with squamous lung carcinoma. We only studied in the context of transformation from lung adeno. And that's about the data that we have. So I wouldn't be able to tell you the transcriptomic status in these lung squamous carcinoma. We haven't done the study yet.
I just want to make a comment that Sloan Kettering and New York Hospital is my stomping ground. I did my postdoctoral training at New York Hospital. So this was during the era of Bob Good when he was the director of Sloan Kettering. So.
Great. I mean, that's amazing. I've been only here like a couple of years, but it's such an amazing place to be working with, and the crosstalk between basic scientists like myself and doctors. It's just the most impressive thing that happens at Memorial. And that's so important for conducting research like the one we do.
Yeah. Okay. Thank you very much.
Triparna, do we have time for one last question from our audience? Okay, so from Dr. Song, the STK keep1 double KO cells proliferate faster. How did you normalize when you did the CRISPR screen and in vivo work? Did you collect cell pellets at different time points?
Yeah. That's a brilliant question. That's amazing. Yeah. So we did a lot of QC. We went back a lot. That's why I showed you 16 doublings. It wasn't a time, because when we wanted to decide the endpoint, like 21 days or 23 days, you're right, because the double knockouts, they proliferated way, way, way faster. So they reached, by 21 days, they were already a lot higher. So what we did was we did 16 doublings. And we collected whenever each cell line reached 16 doublings. So we did not denote the time, we noted the doubling. And the model that was then considered for next generation sequencing would have undergone 16 doubling. I hope that answers the question. But as you said, I mean, Pierre has my email. I'm happy to share my email. If any of you have any follow-up questions, or if you need the druggable genome library or anything, we really would like someone else to test it for us and say how good the library is. So I'm very open to even you all can email me about any follow-up questions, queries, concerns that you may have.
Wonderful. Again, Triparna, thank you so much for your excellent presentation. And thanks to all of you for attending this seminar. We will keep you posted for our next seminar, which is going to be scheduled for the beginning of April. And we'll follow up by email about that. So thanks to all again. I hope you enjoy the rest of your day, and we'll be seeing you soon. Thank you.
Thank you.